Hi, I am Mrs. Sloan, and this is part 3, video 2, but part 3 in a series on control of gene expression for my honors biology students. So video 1, we discussed part 1, which was prokaryotic control of gene expression, and part 2, which was eukaryotic control of gene expression. And then today what we're going to talk about is failure of control of gene expression. And what we call that is cancer. So let me get a pointer right here. So down in the descriptor of this video, I have notes, and the notes contain a link to this presentation you're seeing right here, if that would be helpful for you. The notes are 2-column notes. Column 1 is a scaffolding, and I'll help you fill that in. Column 2, I encourage you to throw in pictures and images that are helpful for you. So for my students, we are on 25.2, cancer, a failure of genetic control. Well, let's talk about what a normal cell does. A normal cell exhibits what's called anchorage dependence. It's got to be hooked onto something before it will ever do mitosis and make more cells. Are there exceptions? Sure, red blood cells, white blood cells, but most cells need to be anchored to something. Another characteristic of a normal cell is density-dependent inhibition, meaning if it's crowded, if there's another cell right next to it, it will not undergo mitosis. If there is a vacancy, here you can see they've removed some cells out, then it will undergo mitosis and fill in the gaps. So for your notes on your introduction is normal cell division, A. Density-dependent inhibition is crowded cells stop dividing, and anchorage dependency, you have everything for that. So here, what happens when you don't have anchorage dependency? What happens when you don't have contact inhibition? You can get an abnormal growth of cells known as a neoplasm or as a tumor. Those are pretty much interchangeable. So a neoplasm is an abnormal growth of cells. And you can have two types, both. You could be benign or you could be malignant. Now, benign means currently it is not harmful. It could become malignant, but currently it is not. It's usually encapsulated and it's surrounded by normal tissues. You could go in and get it removed if you detected it, and then you would be all right. If it's malignant, it means it's spreading. It has broken out of the confines of that tissue, and it may even be metastasizing and getting to other tissues of your body. So on your notes for tumor, mass of abnormal cells within otherwise normal tissue. A benign tumor is not cancerous. It's encapsulated. It has not invaded adjacent tissues, but if it's malignant, it has the ability to spread. Now, how does it spread? Well, there are two superhighways in our body, the circulatory system and the lymphatic system. And basically, it's like a subway system. And so where you have cancer, it can come out of the area it is in. If it is able to, it has to gain a mutation in order to escape in this way to get, like, for instance, you have cells here called your basal lamina that separate two different tissues. And if it has the ability to digest that barrier, it can get out and jump into either your lymphatic or your circulatory system. And this would be like at a subway stop, you get on and you ride that circulatory system and jump off at another tissue. And here we can see what happens when that, so here it's jumped into the circulatory system and then it's landed here in a secondary tissue, and it's setting up camp there. And remember, cancer is going to interfere with whatever that tissue is supposed to be doing. When you hear about stage 1, stage 2, stage 3, stage 4 cancer, what they're telling you is where it has traveled to. For instance, testicular cancer originates in the testes. If it's metastasizing, the next place it'll set up camp is in lymph nodes around your groin region. That would be stage 2. Stage 3 would be if it sets up camp in your lungs, and then stage 4 would be when it goes all the way to your brain. So on your notes, whenever it says metastasis, cells separate from the malignant tumor, enter a blood or lymph vessel, and travel to other parts of the body. So cancer is a cellular growth disorder that results from mutations. You have a mutated gene, so it codes for wrong proteins, and these wrong proteins are telling your cell cycle. Remember cell cycle? Do you remember G1, S, G2, mitosis, cytokinesis, right? It's telling you go cell cycle when you shouldn't. And so they're out of control. Okay, and so on your notes for cancer, the word that you need is mutations of genes. That's number 3 in your notes under the introduction, that regulate cell cycle, and you have loss of control. Loss of control. And here's that cell cycle for you, right? So you have interphase, which is G1, S, and G2, and then you undergo mitosis. So it's defying those regulatory mechanisms, and then it has the ability to invade other cells, other tissues, other organs. That's when you have cancer. Now, the process of developing cancer, remember, genesis means the beginning. So the beginning of cancer is usually gradual. It can take years, decades. If you're exposed to a mutagen, it could happen fairly rapidly, like Chernobyl. You will, that big input of radiation can immediately mutate your genes, and you have cancer, but usually it takes decades. Like smoking, like you could smoke in your 20s and end up getting cancer in your 40s, 50s, or 60s. I think that if you smoked today and tomorrow you got cancer, there'd be a lot fewer smokers. And by the way, vaping is bad too, so just stop it. Okay, so carcinogenesis on your notes is the development of cancer, which is usually gradual and can take decades. An exception to that would be retinoblastoma. It's a single mutation, and that's cancer of the eye. So let's start looking at some of the characteristics of cancer cells. This is abnormal here to have these extensions. They usually have blisters on them called bleebs. Here are 3 white blood cells trying to take this cancer cell out. So comparing and contrasting normal to cancer cells, what they look like underneath the microscope. So cancer cells lack differentiation. They are not specialized. Lack differentiation, they are not specialized, and they seem immortal. And we'll talk about that in just a minute. They have abnormal nuclei. So here you can look at the nucleus right here. They have multiple nuclei, and this cell has 2. Large nucleoli, very coarse chromatin, and very little cytoplasm in these cancer cells. So on your notes, let's say, abnormal nuclei is what you have for your notes. Some other characteristics of it is normally when your DNA is damaged, right, you will repair it. You'll either kill those cells or fix those cells. If you can't fix them, you'll kill them. But in cancer, these damaged cells are allowed to proliferate and maybe even gain other mutations, and mutations that allow them to metastasize and spread to other areas of your body. So these cells should undergo apoptosis, but they're not. So remember, apoptosis is cellular suicide. So number 3, do not undergo apoptosis, even though abnormal cells are aged DNA. Normally cells enter the cell cycle about 50 times, and then they're done. All right, so this can lead to a tumor. So a tumor is a result of lack of density-dependent inhibition or anchorage dependence, and you start to get this conglomeration here of cells. So do not display density-dependent inhibition or anchorage dependence So now they want these cancer cells then are going to spread to other areas. So, and we already talked about metastasis. So just add this. This is the second time I'm talking about it. So metastasis is new tumors form, which are at a distance from the primary tumor. And this tells me that I gave you that definition before. So let me help you. Go back to introduction to D, where it says metastasis. Cells separate from a malignant tumor and enter blood or lymph vessels and travel to other parts of the body. That's what you need to do up and to. Sorry about that, my excellent students. Okay, now another thing, besides spreading to distant tissues, another trick they have up their sleeve, if you have this primary tumor right here, now, remember, your cells need oxygen and nutrients, and they need to get rid of waste. You start having a massive growth, it's not going to have the blood supply that it needs. But tumor cells are super tricky because what they do is they secrete through chemical signaling to your blood vessels and say, come here, little blood vessels, grow to me. So your own blood vessels will receive signals from the tumor to grow towards it, which then supply it with all the nutrients and oxygen it needs and to get rid of its waste. And then it could gain further mutations, would allow it to spread to a secondary site, and that is called angiogenesis. So when a tumor cell acquires additional mutations that allow them to direct the growth of new blood vessels to the tumor, new blood vessels to the tumor. So here, this might be a good thing to pop into your notes as a picture. You can just compare and contrast cancer cells to normal cells. And I would just hit pause, screenshot, and throw it in your notes. All right, now, why does this happen? Okay, let's get to the genes of it, the genetic origin of it. There are two categories of genes that play a very important role in your body. Number one, proto-oncogenes. Now, you see proto-oncogenes. What kind of doctor do you go to if you have cancer? You go to an oncologist, right? So these are proto-oncogenes, genes that could end up contributing to cancer. And it's not that these are normally bad genes, but they're in a position of power. So for instance, if you have a general and this general goes bad and starts directing all of its troops to go bad, because it's in a position of power, it can influence so many other things in your body. That's what proto-oncogenes do. When they mutate, they can become oncogenes, and those are genes that facilitate the cell cycle, and I'll tell you more about that in a minute. Now, if you have an out-of-control general, what you can do, and I'm not saying this is what we should do politically, this is just an analogy, okay, is you have these tumor suppressor genes that are like assassin genes. So if the proto-oncogenes become oncogenes, they fail, normally your tumor suppressor genes would stop them, take them out, take out the bad general, and then you won't end up having cancer. So the analogy I like to use for this is driving in your car. So if you're driving in your car and you're just driving along normally, and then your friend reaches over and just like steps on your foot on the gas, and you're headed towards an intersection, and you're like, what are you doing? We're going to an intersection. This would be what, when a proto-oncogene, your friend has mutated to an oncogene, you're stepping on the gas and you're headed right to a crash. But what tumor suppressor genes do is they help you put on the brakes so you can stop and not get in that accident. When these tumor suppressor genes fail, it's like your brakes failing and you crash. So two things have to happen for you to have cancer. Your proto-oncogenes have to mutate into oncogenes. That's like your friend stepping on your gas pedal and you're like, no, don't do this. And your brakes have to fail, that are always there looking out to make sure you don't crash. All right, so let's talk about this. Proto-oncogene, if it mutates into an oncogene. It will cause cancer by coding for proteins that will facilitate the cell cycle. They're like, go cell cycle, go cell cycle, go. And this would be like being unable to take your foot off the gas of the cell cycle. You just keep moving forward. So on proto-oncogenes, for your notes, these code for proteins that promote the cell cycle and prevent apoptosis. And I said, like a gas pedal on your car, it's a stimulatory pathway. Now, how could that happen? Mutations, right? You could be out in the sun too much, no sunscreen. Those UV radiation could cause a gene to mutate. And so instead, so here you can see a proto-oncogene in a position of power. Normally it's good. But if it mutates, it becomes bad, an oncogene, and now it will trigger the cell cycle even if it isn't supposed to. So it is a stimulatory pathway. And my friends, how many proto-oncogenes do you have? You have over 100 potential oncogenes, over 100 potential oncogenes. Examples of those are, here, I think I have a picture for you right here. Let me go. Yes, the RAS. The RAS gene codes for the RAS protein. So I always remember this because RAS is like stepping on the gas. Okay, so normally it would need to be triggered by a growth factor binding to a receptor, which then triggers this RAS protein, which then triggers your DNA and gene expression like go cell cycle go. But if you have a bad RAS, even if there isn't a growth factor, it's still telling your DNA, hey, let's go, let's make more cells. Okay, so that's the RAS gene family. It's associated with lung, colon, pancreatic cancers, leukemia, lymphoma, and thyroid cancers. That's a lot. And then I'm sure you've heard of another one, the BRCA1, BRCA2 genes, breast cancer genes that promote tumors and cell growth in breast tissue. So that is another one. That is a proto-oncogene. Okay, now let's talk about your assassins. Okay, you have a tumor suppressor gene that codes for proteins to stop the cell cycle, try to fix it. If you can't fix or repair what's gone wrong in your DNA, then commit apoptosis. These are your brakes on your car. Okay, these code for proteins that inhibit the cell cycle and promote apoptosis. So if you have, brake pads, you've got your wheels, right? Your tires. If you have one bad brake, you can still be okay as long as you have a good copy. But if both your brakes are bad, that's it. So if you have a proto-oncogene that mutates to an oncogene and says start going cell cycle, you have no way to put the brakes on that. Now, one that you want to know about is, remember when we talked about control of the cell cycle and we have checkpoints. So here, following G1, you have a checkpoint, and this is all about the P53 gene. And this gene will put the brakes on your cell cycle if there's any damage, fix it. If it can't, here, let me show you another slide right here. Okay, if there's any, like here's some UV light, you've got damaged DNA, this alerts, you get the P53 protein, it will either fix it. If it can't fix it, then the cell will commit apoptosis. If your P53 gene, which makes those protein breaks, if it is damaged, then you have no brakes. You have no brakes and no way to control that. So there are about, on your notes, there are about 6 identified, but the P53 is involved in almost half of all human cancers. If we could figure out a way, right, through CRISPR to fix our P53 genes if they're broken, that would take care of half of all cancers out there. So what are the causes of cancer? So one is heredity, right? You could inherit a bad copy of a gene from one of your parents, right? So that's number one. And then number two is your environment that you are in. So there are chemical carcinogens, smoking, right? Smoking, vaping, bad. There are so many carcinogens, and then you're more likely to mutate, exposing yourself to sun that without any sunscreen, that could lead to cancer. Unhealthy habits, obesity, more likely to lead to cancer. And then mutagens within your environment. So on your notes, 1, 2, and 3, chemical carcinogens like smoking, radiation like the sun, viruses that you could be exposed to like the HPV virus and cervical cancer. Those are all things that could lead to cancer. Now, diagnosis and treatment. What can you do about it? Well, to diagnose it, you can have screenings like mammograms, pap smears or screenings. So on your notes, routine screening tests, colonoscopies are screenings. You might screenings, colonoscopy, mammograms, and a pap smear. You can also, there are blood tests that you can take, tumor marker tests, where you are looking to see if certain proteins are getting generated and put in your blood supply. Then that could tell you if you have potentially cancer. So tumor marker tests, blood. Example, they check for PSA for prostate cancer. Okay, that would be in males. You can have genetic tests for colon, bladder, breast, thyroid, or melanomas. There are genetic tests to see if maybe you have inherited that. And then next, what you need to do is confirm that diagnosis. So if they believe that you have a tumor in your breast, then they will go and do a biopsy and check to see if there are, and they could recognize underneath the microscope if there are abnormal cells, like what I talked to you about earlier, and then go and try to remove that tissue. There are different imaging techniques that can be done in order to see if there are any abnormal growths or neoplasms as well. So 4A, biopsies allow for removal of cells for examination, and then the imaging techniques, radioactive scan and ultrasound, we want to add in there, and ultrasound. And then as far as treatments go, There are standard treatments, surgery, radiation, chemotherapy, monoclonal antibodies. These are all things that can be used to help treat. And then for future techniques, it would be great if we could have cancer vaccines, P53 therapy, something with CRISPR, drugs to prevent angiogenesis. So on your future techniques, cancer vaccines, and then the rest I gave to you. All right, so I know that was a lot to take in. Hopefully you learned a little bit more about cancer, and if you're one of my students, I'll see you in class.